100 clicker questions? That's what I said, right? 100 clicker questions? Okay. Yeah, so today we'll talk about archaeal viruses, big picture kind of thing again here in terms of thinking about what you might want to consider in terms of what you'll be doing this weekend, or not, as the case may be. These are almost all double-stranded DNA viruses, and that's actually not dissimilar from the case for bacteriophage. Most of the viruses of bacteria are double-stranded DNA viruses. There are a few RNA viruses which infect bacteria. There are a few single-stranded DNA viruses that infect bacteria, but the majority seem to be these double-stranded DNA viruses. So far, there are no RNA viruses that have been found that infect archaea, but there have been, and I'll talk about this very briefly right at the end, a single-stranded DNA virus which is infecting archaea. Again, big, ridiculously gross picture here. The Euryarchaea have bacterial-like viruses, i.e. head and tail structures, whereas the Crenarchaea have these absolutely bizarre and wonderful virus morphologies, incredibly unique genomes, etc. And this is a quick shout out again to Viral Zone. A couple people asked me about this. It's a really nice website that gives overviews of the individual virus families. So again, in terms of a slightly different approach of looking at some of these viruses, it's a great place to go and look at some of these things. So we will talk briefly about these Euryarchaeal viruses. This is Euryarchaea and Crenarchaea, by the way, for those of you who are not archaeologists. Those are the two main divisions of archaea. Certainly in terms of the cultivated organisms. There's the Euryarchaea, which are the methanogens, the extreme halophiles, and the Crenarchaea, which are the coolest, are the ones that mostly grow, at least the isolated organisms, at high temperatures and low pHs. So the boring viruses of Euryarchaea we'll cover very briefly because, again, they look a lot like the bacteriophage in terms of their morphology, but also in terms of their genomes and replication. The viruses of Crenarchaea are just really bizarre. And again, that's partly why I'm so excited about them. We'll spend most of our time talking about the Fusella viruses, the spindle-shaped viruses. This is the real focus of what we're working on in my lab right now, and also the mutant viruses from Hell Lab that will be doing their presentations this afternoon and tomorrow afternoon. So come check them out. If you're interested, if you're in the class, you have to be there. Then we'll also talk about some of these other viruses here. We don't have time, of course, to go through all of them. One of the really amazing things about these viruses of Crenarchaea is each of these is classified taxonomically as being a completely new virus family. So the way that we've been talking about viruses so far, generally sort of one lecture per virus family. So this is what, 1, 2, 3, 4, 5, 6, 7. Maybe I should just do a viruses of archaea class. That would be so much fun. And this is actually a too short list here. But we'll talk about some of these in more detail, particularly the Rudiviruses, which are rod-shaped viruses, and then the Ampulla-viruses, which have these really amazing virion morphologies, kind of look like champagne bottles. Maybe crack one last new lecture of the term. And then these Turriviruses, turrited icosahedral viruses that we've talked about a little bit before, but I wanted to talk about a slightly different aspect of these before. So as you can tell, I'm pretty excited about these particular viruses. Unfortunately, most of the rest of the virological world thinks that they're really pretty boring relative to everything else. This is the sum total of archaeal viruses in the virus sphere. Admittedly, a few years ago, but it hasn't changed much. So nobody really cares about these other than people like us. Now, why should we care about viruses of archaea? Well, first, archaea are one of the three main cellular groups or domains of life that are present on our planet. So these are just a few of those archaeal microorganisms here. Bacteria, there's also a huge amount of diversity in bacteria. And when we talk about eukarya, mostly we're talking about animals represented down here and plants here. So huge amounts of diversity, practically nothing known about these organisms and also the viruses which are infecting them. Archaea are everywhere. They're not just present in these extreme environments. Turns out that they're really hard to work with if you find the ones that are not in the extreme environments. And so that's one of the reasons that they've probably been somewhat understudied. They, at least as far as when I'm writing grant proposals, are really similar to eukaryotes and us. And we can find out really fundamental aspects of molecular biology by studying Archaea. Again, this is at least what I always put in my grant proposals. I got really interested in them because they do survive in these really extreme environments. Everything I learned about biology, which wasn't that much, said that life can't survive in boiling acid. But these things do. So how? How can they do that? Evolution, of course, is the right answer. They have a few other really fascinating aspects about their biology. Their lipids are completely different than other lipids. Some of these are actually tetraether lipids, so it's a lipid monolayer that's on the outside of some of these cells. And they may or may not be similar to ancestral organisms. That's why they're called Archaea. The vast majority of archaeal viruses infect sulfolobus, which goes optimally at 80 degrees Celsius and pH 3. So a pretty extreme environment. And then, 90% of the viruses we've talked about so far infect these kind of boring organisms down here. So the thing that got me particularly interested in these viruses was that the virions of these archaeal viruses are just completely different, for the most part, than any other virions that people had seen before. This one we'll talk about a little bit later. The Acidianus bottle-shaped virus really does have this like champagne bottle kind of structure. This one actually looks a little bit like some of the enveloped viruses that we've talked about before. Turns out it doesn't really have much of an envelope. This is the pyrobaculum spherical virus, we're not going to talk too much more about. This one is the Acidianus filamentous virus. The virion here, the scale bar down at the bottom, sorry, my pointer's not working terribly well, is 100 nanometers. So these virions are about 2 microns in length. So if they weren't so narrow, you actually could see them under the light microscope. But the poor cells that they're infecting are only about a micron in diameter. So these virions are twice as long as any of the cells that they infect. How those get produced, really interesting question. How they get in, they have these nano-sized claws at the end of each of the virions that then close down on pili. And that seems to be the way that they do their infection process. This one, STIV, we've seen a few times before. This is one that I discovered in a hot spring in Yellowstone National Park. In fact, the first one that looked at all like most of the icosahedral viruses that we've talked about before, at least viruses with icosahedral virions. This one, of course, is different because it's got a totally bizarre, new, and unique T number. Woohoo. But more importantly, these turrets and also the structures of the capsid proteins, which turn out to be very similar to eukaryotic and bacterial viruses, particularly some of those giant viruses that we talked about last time, tells us something about virus evolution. But what I want to talk about today, more importantly at the end, is these guys have a completely unique way of getting out of the cell. So it's virus escape processes. This boring guy over here, FIM1, is a virus which infects the methanogens. Turns out that this structure is extremely similar to lambda, and the genome is quite similar to lambda as well. And then our favorite, this is the SSV1 virus, the Fusella virus that we're going to spend quite a lot of time talking about later on. But first, to emphasize how boring the viruses of Euryarchaea are, these are just a few micrographs of some of these virions. The vast majority have these head and tail-like structures, again, extremely similar to what you see in bacteriophage. Sometimes they've got these stiff tail-like structures, which is much more like bacteriophage T4. Sometimes they have these flexible structures here, which is more like what you see in bacteriophages like lambda. But there are a few exceptions to this. Turns out that SH1, this is not a great micrograph, but this structure is actually very similar to STIV, and a nice icosahedral structure. Also has an internal membrane, so there's a membrane on the inside of this icosahedral protein structure. And then a few viruses which have these most attractive shapes, like this one. This is, in fact, a 3D printed model of a halobacterial virus. And you'll notice that these two actually do look somewhat similar to each other. Of course, this one's bigger and more interesting. No, the scale is about the same of these two different virions. So there certainly are some differences which are there. And then down here at the bottom are some virions. Wait, little, can you guys see that in the back? Yeah. These have also these sort of spindle-shaped or lemon-shaped morphology. Yeah, Ian?
The ones that look like bacteriophage, do they tend to have the same, like the H2 or the core protein for genome insertion? Do they tend to insert their genome the same way?
So the question is, how do these viruses infect? How do they get their genomes inside the cell? As far as we know, it's very similar, but very few of them have been well studied. So there are maybe 2 or 3 papers on any of these viruses at all. So pretty few and far between. But certainly, if you look at the structures, they look very similar. And if you look at the genomes which are in there, it looks as if they have portal proteins and the typical kind of injection processes that you would expect from a bacteriophage. So you can make those assumptions that that's what's going on there. So this projector is not great here. How many of you recognize that jacket? It's the one that I wear here sometimes. And gives you an idea how long I keep my fleece jackets for. So this is an example of a typical Sulfolobus habitat that I am trying to collect a sample from in Yellowstone. Unfortunately, again, even with the lights out, this projector doesn't do this justice. That hot spring really does look like tomato soup, and it is about the right temperature, 85 degrees Celsius, and a pH of 3.5. I didn't actually get the STIV virus out of here, but it was actually on exactly the same trip in October of 2000 where we collected the samples that STIV was eventually isolated from. But that particular spring actually is kind of an ugly little mud pot, so it's nowhere near as photogenic. Since I promised that we would have clicker questions today, why don't we start with our first one? See if we can get 100% on all of them today. That would be nice. So virions of viruses which infect which of the following have the most known morphological diversity? Euryarchaea, Crenarchaea, E. coli, primates, or amoeba? And no messing with my statistics, Ian. It's like, you got to come up with 5 possibilities, right? Well, actually, you don't really need to, but.
A word in there that I've missed.
10, 5.
He wants you to read.
Stedman wants you to read. Oh no! Shock horror. You don't want me to get my 100%, do you? Really? Yes, it's the Crenarchaea that have by far and away the largest morphological diversity. E. coli probably has more viruses that have been isolated from it than most of the Crenarchaea, although it's getting close now. Euryarchaea, for the most part, are head and tail kinds of viruses. Primates, pretty boring in terms of their morphologies. Amoeba are really cool, but there are only 2 or 3 different ones. So I would say most morphological diversity are definitely the Crenarchaea. Okay, so let's talk about our favorite, the Fuselloviruses. Again, these are these wonderful viruses, and thanks to Angela for making this particular one. This is the 3D printed version, higher tech, but that one I think is much cuter. The main one of the Fuselloviruses that have been studied is SSV1, or Sulfolobus spindle-shaped virus 1. There are now 10 of these that infect Sulfolobus and a couple that are infecting other microbes. The virion itself is about 60 by 90 nanometers. This is just one of these virions down here at the bottom. And they love to stick to each other and also just little bits of cell debris. And so this is just a piece of Sulfolobus which has been broken off of the cells. The genome, which is packaged inside here, is a nice, normal, boring, double-stranded circular DNA, which is great, makes it really easy to work with, right, everybody in the mutant viruses from hell class? But it has one really fascinating aspect to it, which is very different than pretty much any kind of DNA that people had found before. This DNA is positively supercoiled when it's present inside the virion. And that means it's actually overwound, which helps package it, but probably is also important for stabilizing the DNA under the extreme conditions that it likes to live at. If you think about 80 degrees Celsius, usually your two strands of DNA are coming apart. So if you overwind the DNA, it helps those strands stay together. So how this positive supercoiling works, we've got some ideas, but we're not completely sure about it. So this double-stranded circular genome is packaged inside the virion, which is interesting. What other kinds of double-stranded circular genomes have we talked about? Good review for next week. Packaged by acellular histones. Polyoma papillomaviruses are those double-stranded circular viruses. Those get transported to the nucleus as parts of their virion transport processes. How these genomes get inside the cell, we have absolutely no idea. It's a really interesting question. The genome itself, once it does get inside the cell, integrates in a very specific fashion, which should sound a lot like what virus? HIV integrates in a non-specific fashion. What about specific integration of double-stranded circular DNA? Lambda, exactly. So lambda integration happens there. This virus genome is UV inducible. So you shine UV light on it, it over-replicates. What does that sound similar to? Patty?
Lambda.
Lambda, exactly. So bacteriophage lambda. And this particular virus is from Sulfolobus shibata, which is an organism that was originally isolated in the Beppu onsen area in Kyushu in Japan. Particular high temperature, low pH environment. So what is this genome, which is packaged inside this virion? Here's a cartoon of that. If you come to the presentations this afternoon and tomorrow, you'll see lots and lots of images of these cartoons. But when I started working on these viruses, we knew something about 4 of these genes. There's a VP1, a VP3, and a VP2. These are all virus proteins. Again, should sound really familiar. We've talked a lot about VPs. Structural proteins. VP1 and VP3 seem to make up the majority of this structure, the green stuff here. VP2 is a DNA binding protein that we thought was really, really important for positive supercoiling, but doesn't seem to be the case. And then we have a viral integrase. Again, this viral integrase is quite similar to the bacteriophage lambda integrase, has a similar kind of activity. And it turns out that this is the only one of the 34 genes in this genome that is similar at all to any other kind of virus protein. So 1 out of 34. Pretty few and far between. And so there's a big question mark in terms of what the rest of the genome is doing. And this is really, when I started working on this virus frighteningly enough, about 20 years ago. We have learned something in those last 20 years, fortunately. So a couple of ways of trying to learn something. Yeah?
You might be getting to this anyway, but I see it looks like 4 overlapping reading frames there. Is that on VP1? What are the 4 arrows there?
Okay, so the arrows on the inside here, sorry, I should have mentioned that. The broad arrows here, because we'll be seeing a lot of these later on, these broad arrows represent open reading frames. And so we know for VP1, VP3, and VP2, and the viral integrase, that these make proteins. The skinny arrows on the inside are transcripts. And so these are the transcripts which are being made in a, if you look here, sort of bidirectional replication, or transcription, I should say, also should sound really familiar relative to lambda and the other one. SV40, so these polyomaviruses, where you have bidirectional transcription going from one particular region down here. So yeah, but most of these open reading frames here, we had no idea what they were doing. So there are a couple of ways to try and address that. One is genetics, that we'll talk about a little bit later on. But the fun way to figure out what's going on with these things is to go and find more of them. Do a comparative genomics approach. So this is, sorry, I have to turn the lights down again here. Wolfram Zillig, who was really the pioneer of studying any of these viruses. It was his group that found the original sample for SSV1, also looked at some of the viruses that were infecting the halobacteria. David Prangishvili, who was really sort of taken over after Wolfram Zillig retired, unfortunately passed away. And this is his long-term technician who was so scared that she was going to leave the lab that he married her. Collecting in Iceland in 1995. This is me and Blake Wiedenheft, who gave our seminar on CRISPR a few weeks back, sampling in Yellowstone. And I was very fortunate to be able to go to Kamchatka in far eastern Russia to collect some samples in a place called the Valley of the Geysers. And basically, wherever you go and you find the appropriate conditions of low pH and high temperatures, you'll find these viruses. And these are now an overview of where some of the viruses are. And these are just these SSV, these Fuselloviruses that are infecting Sulfolobus. So some from Kamchatka, which is way over here. Those of you who have played Risk at some point know where Kamchatka is. Most people don't. Most volcanically active place on Earth. It's really an amazing place. About 10 times as many bears as there are humans. SSV1 is from here, the southern part of Japan. Also, we have a number of viruses from Iceland, from that trip I showed you with Wolfram Zillig and David Prangishvili and Ingela Holtz. And then I've collected some samples in Yellowstone, and some other people have. And then our main hunting ground is in Lassen Volcanic National Park in Northern California. So low temperature, let's say high temperature, low pH. Why are we going there? Well, because we get those viruses out, and then we compare all of these different viruses to each other. And those genomes, you make the assumption, is if a gene is in every single one of those viruses, it's probably really important for the virus. And if it's not present in a bunch of viruses, it's probably not going to be that important for virus function. And so this is an overview of 11 different genomes here. If one of these open reading frames is in black, it's in every single one of those, which means, it's probably pretty important for virus function. If it's in gray, it's only present in this particular one, which is SSV1. So this gives you at least an overall indication how important these particular open reading frames, putative genes, are for functioning in the environment. But it doesn't really tell you what they're doing, and it doesn't tell you if they're absolutely required or not. So that brings up the other approach, and this is what we're doing here at PSU and also in the lab, which is doing virus genetics. So basically, I think I've mentioned this before, I'm not sure if it's in this class, but certainly in that class that will remain nameless from last term. What geneticists do is they break stuff, and then they try and figure out what they broke. And so that's exactly what we did here. We started out with something that we thought was going to be pretty easy. Turned out to be a lot harder to work with. We developed this technique which we call long inverse PCR. And long inverse PCR is pretty straightforward. What you do is you design primers, these little red arrows here on the outside, that are flanking the gene that you want to get rid of. Then you do PCR and amplify everything except for that gene. And then you put these two ends back together, and in that process, delete a gene. We figured we'd start out with something we thought we knew about. We took the viral integrase gene. Remember, that's the one which is similar to lambda, takes the virus genome, puts it into the host genome, and what we found, rather to our surprise, is the virus seemed to be perfectly happy when we got rid of this integrase gene. Oh, great. Okay. But it does turn out that this particular gene is conserved in all of those other viruses, so it seems to be important in the environment, and a really brilliant, as far as I'm concerned, because I didn't come up with it, it was Adam, my graduate student experiment. What he did is he took two viruses, one that had the integrase gene and one that didn't have the integrase gene, and just said, which one does better? And he found that the one that had the integrase gene won in about 10 generations. So the lacking integrase gene does seem to be important in a real environment, but doesn't seem to be important in a lab. Yeah?
Could it be that some of the other genes are kind of doing the same thing, but not as well?
So the question is, could maybe one of the other genes be doing this integration event and just not as well as the normal integrase gene does? Because again, we don't know what most of these other genes are doing. The answer is it clearly doesn't integrate into the same place in the genome that it would with the other integrase gene. There may be some other kind of recombination that's happening and it's inserting somewhere else in the genome. We don't have any direct data for that.
I was just thinking about the integrase gene. You were saying that it's happy, the virus is happy. Does that mean it's still integrating? It's not, I mean...
Okay, so basically what happens to the virus when it's lacking the integrase gene? We know it doesn't integrate into the place that the wild-type virus integrates. What we don't know is if it might integrate, as your question was, maybe somewhere else in the genome. That we don't know. But we do know that it does seem to replicate as a circular DNA and seems to be able to replicate as a circular DNA. And so it doesn't require integration. So that's a big difference relative to what? Now, lambda actually doesn't have to integrate either. But what viruses did we talk about recently that have to? Retroviruses. Retroviruses have to integrate. Why do they have to integrate? Because they're RNA, true, but why? Why particularly for RNA? What makes that RNA? The cellular machinery. So it's got to be into the genome so the cellular machinery can make that RNA. Whereas in this case, it's DNA, and it's cellular machinery which is making the DNA. There's no DNA polymerase, at least no detectable DNA polymerase in any of these genomes. But that's it. Yeah, Ellis?
Why isn't the fitness decrease observed for 10 generations, you said?
Why in 10 generations? We're not quite sure why the integrase containing does better than the one that doesn't.
Is it just like a numbers thing as a comparison over?
Yeah, so we did 10 generations, we did 100 generations, I think we did 4 generations. I have to go back and look at his paper. And basically you just saw a disappearance of the one that didn't have that integrase gene in it. So presumably outcompeted, but why it was being outcompeted, anybody's guess. Presumably it's a persistence thing. So when the cell divides, if you just have a circular, extracircular piece of DNA, some cells will get it, some cells won't. And those cells that don't have it are going to start to grow better. Again, this is hand waving.
So when you guys were looking at this, was the infection taking place simultaneously in the same culture?
Yes, so we did a competition experiment in the same, they're literally right together with each other.
Not separate over time?
No, not separate over time. These were together, literally competing with each other for the same nutrients, the same cells, et cetera.
Is it possible that the lacking the knockout integrase integrase, picked up integrase from one of the healthy wild type?
So, yeah, the question is, in this experiment, could you have recombination that took place and then replace that virus integrase? If that had happened, it's likely that we would have seen it later on. Now, one of the things that I didn't mention here, but I suppose I probably should, is that the integration site here, this little black bar in the middle of the integrase gene, that's where integration takes place. And so just like lambda, you have a circular, you open up that circle and put it into the genome. When that happens with lambda, the integrase gene is still a complete and functional integrase gene. In the case of SSV, it actually chops this integrase gene into two pieces. So any kind of recombination from the genome is unlikely to be able to repair that deletion that we made. Now, could you have co-infection in a single cell and get recombination? That's certainly possible, but how would you detect that? And that would also lean you towards saying that, yes, plus integrase is way better than minus integrase. So Adam did this, 11 years ago we published this, kind of frightening. Eric Iverson, a much more recent graduate student, basically said, this is a really cool technique. I'm going to do it for as many genes as I can in the rest of the genome. And that's this overview. You'll notice this is a more recent paper down here at the bottom. Was literally published in May of this year, where he took all of these open reading frames, again, these big wide arrows here. Everything which is red or green is something that he deleted with this long inverse PCR. And there are a couple of things to notice here. One is that there are a bunch of reds, quite a few greens, particularly down here at the bottom of the genome. These are all the open reading frames that you can delete, and the virus is perfectly happy. You'll notice that the integrase gene here, those of you keeping track, is actually now red. Turns out that deletion of the integrase makes a difference what cell you're infecting, so there may be something about something in the cell and recombining somewhere else. But there are a couple of things to take home messages here. One is that, yes, you can delete a bunch of genes, but no, we wasn't able to delete all of them. And the reason for that is this long inverse PCR. It took Adam, the first graduate student, about 3 years to get that first deletion. Then Eric got pretty good at it, got it down to about 6 weeks a time. But he started out, it took about 6 months. So it was really kind of challenging to do all of these deletions. So we argue whether it was Eric's idea or my idea. We said, let's try a different way and do insertional mutagenesis. And so all these little arrows on the outside here represent transposon insertions that were characterized in the mutant virus from Hell Lab before people are taking it now. And now there are many more mutants which you can hear about this afternoon or tomorrow afternoon, where any of these transposon insertions are. But now with these transposon insertions and deletions, we actually have we have a mutation in every single one of these open reading frames. And again, sort of take-home message here, which is quite interesting, we can get rid of almost half of the genes, either by insertion or deletion, and the virus is still functional, which is pretty amazing that you can get rid of half of the genes and a virus still functions. But bear in mind, that's functioning in the lab. And whether we were to put these viruses out in the environment or do those kinds of competition experiments that we talked about, which we haven't done, it's not clear whether these viruses would still be able to make it. And there are a couple of particularly interesting aspects here that I wanted to talk about. We could spend hours talking about this, and again, people will talk about them this afternoon for hours, is what's going on up here at sort of the top of the genome. I didn't label these here, but this is the VP1 protein. This is the VP2 protein. This is the this is the VP3 protein. This is the VP2 protein. The VP2 protein we can delete, and the virus is perfectly happy. Turns out that's not terribly surprising because a lot of viruses don't have VP2 in them. But VP3 and VP1 are present in every single one of these viruses. So what's going on with this? Sorry to click through here. Turns out that if you delete the VP3 protein, you get strange-looking virions. They're much more elongated, kind of more sausage-like than nice lemon-like particles. So probably the VP3 protein is important for making sure that you get the right shape here. But apparently, the virus can get away with changing and actually literally getting rid of this VP3 protein and still making infectious virus. And so these particular particles are infectious. Whether they're as infectious as this, we really don't know. But we're really excited about this mutant because hopefully, again, as good geneticists, we broke something. Now we see an effect after breaking it. Can we maybe figure out what it's actually doing? And so that's the process that we're really trying to follow up right now in the lab. So as an overview of this genetics and genomics stuff, the genomics is look for genes that are present in all these viruses from all different parts of the world. Again, this is the fun part. You get to go and collect samples all over the place. Drag the lab down to Lassen Volcanic every year. And just look at the genes in the genome. Only about half of those genes are present in every single one of these viruses, and it turns out that about half the genes you can get rid of, and the virus is still functional. They're not exactly parallel to each other. Again, the viral integrase gene, the VP3 protein gene, but they're pretty similar to each other. This is pretty amazing. That's why we ended up on the cover of the journal Virology, is that this is really bizarre that you can get rid of half of the genes and the virus is still actually pretty functional. The other thing that we're really excited about is that these VP3 mutations make very different virus structures. But how do we know about these virus structures? Turns out that this was about as good as it got as far as virus structures until about 3 years ago when I got together with Mark Murray, who's at the UT Medical Branch. I don't know if any of you remember, I showed the picture of the BSL-4 lab right in the path of the hurricane. That's right where he works. And there's the Ebola virus micrographs. Everyone's seen the really beautiful Ebola virus. How do you take pictures of a really nasty pathogen? You have an electron microscope at a BSL-3 lab. And that's exactly where those pictures were taken. So this is actually a slightly different microscope that was used here. Cryo-electron microscopy. We, and I say we, it's the group down in Texas, took 1,000 pictures like this in cryo-EM, averaged them together, and we came up with this nice three-dimensional structure here. A couple of things about that three-dimensional structure that are really fascinating. I'm only going to talk about one of them, and that is that the postdoc who was working on this, Matul Saha, had this idea that this structure here, which should look like kind of a stretched icosahedra. Remember, icosahedra are nice, normal, quasi-symmetric kind of particles. This one looks like it was a normal icosahedra here, but then got really stretched out at either end. This is what's called fused fullerene cones. This should sound really familiar because we talked about the nucleocapsid of some other virus not that long ago, which also has this sort of structure. So people used to think that this structure was really bizarre and you didn't find it very often. And this, again, overall architecture. But if this is true, and we still need to show that this is true, but it's a pretty good approximation to start with, is that the structure of SSV might actually be architecturally pretty similar to the nucleocapsid of HIV, and then maybe this could be much more of a fundamental way that you can put virus particles together. So it's not just icosahedra and helices. You can actually have this sort of, I don't know, intermediate between those two, where you have something that's generally icosahedral, but not with that true quasi-symmetry. And it's basically by adding a whole bunch more hexameric subunits between each of those pentameric subunits, which you can see here in green. So, oh yeah, Ellis.
How does that fit into like the VP3 deletion? I mean, do you expect there's more subunits?
Right. So how does this fit together with that deletion of the VP3? If you look carefully at the ends, and I'm not going to go back to it, of the VP3 structures, basically what seems to have happened is it's been stretched. And you could imagine that this nice rounding up of the structure is due to VP3, and that stretching is when you don't have VP3, which is there. And so that's the hypothesis that we're trying to address right now. And so presumably you would expect to see VP3 right around here in virus particles and not so much at either end. The ends look extremely similar in those VP3 deletions. Yeah?
Sorry, just going back just a little bit. You're saying that half of the genes aren't really required. Is this something that you see or that is presumed to be seen more in archaeal viruses? And if so, why? Is it because that they aren't maybe as able to, or they don't have as sophisticated a system to repel the viruses, and so the viruses are allowed to kind of be a little bit more sloppy or laid back and, you know?
Laid back viruses, I love it, yeah. So basically the question is sort of what's an explanation for the fact that you can get rid of so many open reading frames and the virus is still functional? And is this an archaeal virus specific thing? Genetics have been done on 2 archaeal viruses. This one and the other one that I discovered, and way more in this one than any of the other ones. So we can't generalize for these archaeal viruses, that we don't know. That's the first answer. And the second one is sort of, it's the why question. And the answer is, well, evolution. But quite why that is, we really don't know. And it's a very interesting question. I think to address that will have to be some of these evolutionary kinds of questions about how well they do relative to each other. One thought, you remember these are all from viruses from various different parts of the world. Maybe they can infect some host cells, like the Japanese viruses love the Japanese hosts, or the Japanese hosts have developed defense mechanisms against the Japanese viruses. Can you look at some of these kinds of structures? So we actually tried to do that a couple of years ago, and it turns out it's kind of a mess. There's no nice, easy way of saying the virus from Kamchatka can't infect people in Kamchatka, or should it people, Sulfolobus from Kamchatka, but can infect other ones. It's this whole mismatch of things. And so one of the things that we're very interested in is now looking at what's happening with these various different virus mutants, if those then are different in terms of what they can infect. So we suspect that there's some of that going on as well. And it may be that there are differences enough in the hosts that it's these differences in the virus genomes that are important for these different hosts. Yeah?
Can these viruses infect, like if you get it from Kamchatka and take it here, can they infect Sulfolobus here?
Yeah, so the question is, so this is the host range question. Curiously enough, these are the 3 slides that I took out last night. So it turns out that the Kamchatka virus is really nasty. It can infect practically everybody. And that would be Sulfolobus from Iceland, Sulfolobus from the US, Sulfolobus from the original isolate, which is from Italy. Basically infect everybody. SSV1, the one that we work with, is actually kind of a wimp, relatively speaking. It can only infect relatively few different hosts. So it comes originally from this Japanese one. It can infect one of the ones that we have in the US, one of the ones from Italy, but none of the Kamchatka isolates, none of the Icelandic isolates, et cetera. So yeah, but there's no obvious difference there that we can say definitely is going to be this. But now we have some mutants in this guy where we can start to make some of those experiments and check them out. Okay, so I basically talked about this already. Everyone clear on SSV1? More SSV1 questions? If not, I'll ask you one. So, according to Stedman and coworkers, the structural architecture of SSV1 is most similar to that of bacteriophage MS2, bacteriophage T7, mimivirus, HIV1 nucleocapsid, or SV40. Should say the SSV1 virion, not the genome.
Can you have architecture in the genome?
People talk about genome architecture, yeah. So also genome synteny, so the order of the genes or the orders of the transcripts. Yeah. Yeah, we have a little place next to the lab we call Mordor. So artificial hot springs, so high temperature, low pH.
That's where the mutant viruses repel.
That was my graduate student who came up with that.
Yeah,
so
that's great.
I can't claim to have come up with that. So, but it's too good not to steal.
Yeah. Keep it.
10, 5. I keep hoping for 100% on these, but we're getting to 94. So last one was 90. Now we're at 94%. The next one will be 100, right? If we even get to that last clicker question, we shall see. So yes, it is the HIV-1 nucleic capsid, this fullerene cone pulled icosahedra kind of structure, which is there. One thing that is, again, getting back to the genome architecture question that came up here before, if you're thinking about how the transcripts are organized, that would be rather like what's going on with SV40. Okay, so now we're going to do I wanted to say 30,000 foot. This is more like a 50,000 foot overview of some of these other viruses. I mentioned the Lipothrixviruses already. Lipo being lipids, trix being a thread, and come on, computer's giving me a hard time. It can't do this yet, so one more lecture to go. So these are these long filamentous viruses. They have double-stranded DNA genomes. They have inverted terminal repeats at the end, so they're not circular genomes. These are linear ones, and actually extremely similar to the pox viruses and the giant viruses in terms of their genome structure. Very, very similar from that point of view, except that these guys actually have telomere-like structures, so these 6 base repeats that are present at the end of the genome. No obvious case of a telomerase present in these genomes, but that's what these genomes look like. And again, they've got these claw structures. You can see if I get my pointer to finally work here, it's not. But down here at the bottom, these claw-like structures. This is again from viral zone. And these close on pili, which is probably the way that you're having interactions that take place with these. The Acidianus bottle-shaped virus, this is also known as ABV. These have probably the most amazing morphology of any virion that I know of. They really do look like these bottles. They have a ring of filaments around the end here that they conveniently colored in red in this picture here, and the cartoon is down here. They kind of look like birthday candles at the end. Turns out that this is probably not the end which is interacting with the receptors. It's this end down here. The genome seems to be packed around this cone-like structure. Again, it's a double-stranded DNA genome, but unlike these other viruses, it's linear but has a protein priming at one end. So you've got a protein covalently linked to each end of the genome, which is really similar to what viruses we've talked about so far? Polio, VPG, that's an RNA virus, but for DNA viruses. Herpes viruses actually are not protein prime. That's the rolling circle replication. Adeno, exactly. Once they got the terminal protein. So protein prime replication. Yes? What's the proposed function of the birthday candles? There's really no proposed function for these birthday candles. They're just really fascinating that they're there. But what on the heck they're doing? Turns out that this is a very difficult virus to study. You get very low numbers of virions, and growing the cells that they're associated with is really quite challenging. So turns out that this is you'll notice that the latest publication here was in 2005. And there's really been nothing since then that's been published there. It does have a DNA polymerase gene in it, which again seems to be important for this protein primed replication. Yeah, Angela?
Oh,
So what's the environment that this is found in? So the host for this is Acidianus. Acidianus is a very close relative of Sulfolobus, and it comes to give you an idea, the acid part at the beginning. So again, it's a very low pH environment, and turns out also these are hyperthermophiles. They're found in basically identical locations to Sulfolobus, and where this particular virus was found was actually in the same location as Sulfolobus sulfotericus. The very first Sulfolobus sulfotericus was ever found, just outside of Naples, Italy. Again, just a horrible place to have to go and collect samples from. Okay, so this is, these are the fascinating parts about these viruses. How they get in, probably through the cork-like structure at the end here, but no one really knows exactly how these guys are interacting. There may be, getting back to your question, Ellis, there might be some interactions here between these filamentous things that you see in the EM, which might be pili, but it does seem as that the infection process is going on through this end of each of these virions. There are some more pleomorphic kinds of viruses that you find in some of these species. This is the Pyrobaculum spherical virus, very much an enveloped-like virus. Probably the main important thing here is that almost all of the open reading frames are in one strand in terms of how they're being organized, but much more importantly is that none of these open reading frames match anything in the databases. So this genome has zero similarity to anything. So a lot to do, but challenging in terms of trying to figure out how to do it. Another, again, sorry about this 50,000-foot overview here. One of my other favorites of these, so I've got SSVs, these are the ones that are closest to my heart, but there is the Acidanus bottle-shaped virus, which has this really fascinating overall structure. But then this Acidianus two-tailed virus, again, Acidianus infecting very similar kinds of microbes as Sulfolobus, high temperature, low pH environments. These guys start out looking like a little SSV, actually somewhat larger than the SSV. But if you just take these particles, which are being produced, the virions that are being produced by the cell, and incubate them at high temperature, they grow these tails on the outside of the cell. But wait a minute. No, they're not metabolizing, right? So how on earth are they having this growth taking place on the outside of the cell? We saw this really briefly for HIV, where you have the protease that changes the structure a little bit. But nothing like this kind of massive change of structure. And it turns out that this change of structure here is absolutely required for infection. So these particles are infectious, these particles are not. And what seems to happen here is that if you look at this, it's a little hard to see in this image, it turns out that the volume of these virions actually doesn't change between here and here. And what happens is there's a protein here in each of these tails that starts out in one conformation, but at high temperature changes its structure and then starts to form these filaments. Actually, not unlike what happens with some of the amyloid proteins that have two different structures under different kinds of conditions. It really looks as if this is another example of formation of a particular protein here and then a conformational change here, which is absolutely required for infectivity purposes. I mentioned right at the beginning that mostly double-stranded DNA viruses. Turns out that's not completely true. There's an Reropyrum coil-shaped virus. This is also that's a bit hard to see here, the virions. But these are virions that look like they're all wrapped around just one continuous stretch of proteins with these little projections at either end of the micrographs. These organisms grow at 90 degrees Celsius, where you'd expect single-stranded DNA is not going to be happy at all. But it turns out that they are single-stranded DNA genomes, and in fact, the largest single-strand DNA genome ever to have been found, almost 25,000 bases of just linear DNA. And it's a circular DNA, actually probably replicates in a very similar way to the circoviruses, the things that George talked about a couple of weeks ago. I want to finish up talking about the Reropyrum. These are probably the second best studied of the viruses of Archaea. These are rod-shaped viruses. They're pretty stiff rods. It looks a lot like tobacco mosaic virus. Does have a couple of tail fibers at the end, easier to see in the cartoon down here at the bottom. Many of these have been found. This is just SIRV1 and SIRV2. SI, by the way, stands for Sulfolobus islandicus, found in Iceland originally, rod-shaped virus 1, rod-shaped virus 2. Unlike the case for tobacco mosaic virus, which of course is a single-stranded RNA virus, these are double-stranded DNA viruses, and these genomes turn out to have different lengths, and it turns out that the virion also has different lengths depending on the length of the genome. So if you have a shorter SIRV, you have a shorter virion. If you have a longer SIRV, you have a longer virion. And just by seeing that, you can make the assumption that the assembly is going to be a lot like what happens with tobacco mosaic virus, and that each one of the subunits literally binds to your nucleic acid as you move along the genome. Also, similar to what we saw with the Sulfolobus islandicus filamentous viruses, also pox viruses, we have these closed hairpin ends with inverted terminal repeat structures. So replication, and there's been some data on this, does look like it happens very similar to poxvirus replication, which is also very similar to what you see in adeno-associated virus replication. So it's a nick that happens in the genome. That nick then gets extended. You end up with concatameric structures, which then get put back together. These have very high mutation rates as well. Also an interesting aside, probably because of modifications to the cellular DNA polymerases. But the main reason I wanted to talk about this is that this structure, very recently, or just a couple of years ago now, 2015, they were looking at the structure of each of these virions, and particularly how the capsid protein is interacting with the DNA. And this is, again, it's a cryo-EM reconstruction of this, but the main big surprise here was that the DNA that was being packaged was A-form DNA. Everyone always thought this, B-form DNA. It's what Watson and Crick came up with. That's what most of our DNA looks like most of the time. Packaged in nucleosomes, et cetera, is mostly B-form DNA. Turns out that in SIRV, it's completely A-form DNA. So we had positively supercoiled DNA, which is what you see in SSVs. SIRV, Sulfolobus, also high temperature, low pH environments, has come up with a different way, again, why? Evolutionarily, for packaging its DNA, which is A-form. And A-form seems to be more stable in terms of keeping your genome to be able to package it appropriately. So with that, we'll have one more clicker question. So everyone can get 100%. I'm hoping for this 100%. Which of the following viruses has a bottle-like virion morphology? ATV, ABV, SIRV, SSV1, or STIV? And no, I'm not going to spell out exactly what all of them are. Glug, 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 glug.
I'm not going to read into that at all.
It's that tricky Stedman guy. Trick questions.
I think it's this one. So it must be the other one.
By definition. Think about it. Last clicker question of the term. Oh. So sad. So sad. And lo and behold, drum roll please. Yay! Okay, we did get our 100% here. So, before everyone leaves and runs away, I did want to show you one more thing. And we've got a minute and a half left. So, STIV, totally cool, T equals 31 structure, conservation of structures, tells you something about antiquity of viruses. But it turns out that these viruses and the SIRV viruses come out of the cell through these really funky pyramid structures. So also known as VAPs, or virus-associated pyramids. These are structures that form from a virus protein that when lysis takes place, they open up, kind of like petals of a flower, and release the STIV virion, the sulfilobus turridic virus virion. Turns out there's an extremely similar protein in the SIRV, the rod-shaped virus genome, and this is one of those cells that has been infected by those. The reds are the viruses, and these yellows are those virus-associated pyramids which have opened up to allow those virus particles to come out of the cells. Truly amazing and unique way that archaeal viruses have come up with, again, evolutionarily, to get out of the cells. So with that, thanks for a great term. Make sure you fill out all of your forms. See you on Friday, maybe, and definitely on Tuesday.